In a previous video, titled Electrophoretic Separation of Proteins, we demonstrated how a complex mixture of proteins could be separated using gel electrophoresis. In this video, we will show you 4 different gel staining methods for visualizing the location of the proteins in the gel.
Hi, I'm Sean Gallagher from UVP, working with the Proteomics Center here at the Keck Graduate Institute. Today I'll be showing you a number of techniques for visualizing proteins after separation by electrophoresis. This includes Coomassie Blue, silver staining, SYPRO Orange, and SYPRO Ruby. So let's get started with Coomassie Blue first.
The detection of protein bands in a gel by Coomassie Blue staining depends on non-specific binding of a dye, in this case, Coomassie Brilliant Blue G250. In this method, proteins separated in a polyacrylamide gel are washed using high-purity water. The location of the proteins is then detected using a water-based Coomassie Blue stain. The detection limit is 0.3 to 1 microgram per protein band. To begin, start with a polyacrylamide gel in which proteins have been separated by electrophoresis. While agitating slowly, place the polyacrylamide gel in a plastic container filled with 300 milliliters of high-purity water. Leave for 5 minutes, discard, and repeat 2 more times. After the third wash, pour out the water and cover the gel with Coomassie Blue staining solution.